to get an alternate perspective. All right. Well, I hope y'all had a good weekend. We did get the lab grades uploaded today. I finally finished grading them this morning and got everything posted. This first lab exam, it went how it usually goes, right? I had a few people get A's. I think the high score in here was a 93. So if that was you, well done. Had a few B's, had a whole lot of C's. And I had several people in the high 60s, which means like you're just right in striking distance of a pass on that, right? Had it been any other day, you could have gotten 1 or 2 or 3 extra points and gone ahead and gotten a grade above a 70. And among all of those grades, I typically saw the same type of mistakes. Um, Usually it was silly stuff like we didn't read the actual question, like the picture popped up and you just wrote down the name of the tissue and didn't read that we were asking you for a function or location or something like that. Um, some some folks did not give me the full names of those tissues, right? You left off connective tissue proper or muscle tissue or epithelium at the end of the name. So usually, in most cases, you still get half credit. There are a few cases where you don't get any credit at all, depending on how much of the name you leave off. And just some things, some wrong answers that were in the right universe, right? You identified an epithelial tissue, it was just the wrong one. You identified a connective tissue, it was just the wrong one. And then maybe the associated location was also wrong because you didn't have the right identification. So a lot of good news there. And then for the grades that were substantially lower in the 50s and lower, I don't know if y'all just had a bad day. I don't know what happened. Um, there, I want to help you more. All right, I want to help you be successful. If you want to come look at your exam, you can find me during my office hours, or we can set up a time outside of that for you to come in. We can go over the exam one-on-one, like just what happened. Okay. Um, Sometimes the answer is pretty obvious and everybody knows it before we walk into the room. You know, we got busy, didn't study enough. You know, I didn't know how to study. This is the first time I've taken a test in here. I really had no idea how it was going to go, and I just looked at all the wrong stuff, right? Or I've had a student say, you know, I studied all the lecture materials instead of the lab materials. And in Unit 1, that's really painful because there's almost no overlap between the two. So, um, but anyway, I'm happy to meet with y'all one-on-one, let you go over your test with the answer key. We can talk about, you know, why you got half credit or why you got no credit on a question and sort of try to build a different approach for studying in the future. Now I know who I'm talking to, right? I'm aware my audience is my 5 p.m. class during the week. Y'all got full-time jobs, you got kids, you got the whole thing going on. Everybody's a superhero for just showing up and giving this the good old college try, all right? Doing this class, even if it's the only class you're doing after a full-time job, and then leaving here and going and taking care of your family and then trying to find time to study, right? Y'all are doing it on hard mode for sure. All right. Um, this is if you had a bad day last week, this is one test, right? So mathematically, there is nobody in here that still couldn't get an A in the class, right? Nothing is gone yet. So I don't want you to beat yourself up. Let's try to figure out what we need to do, right? And that is you and me, because I will help you as much as I possibly can. So in that vein, when I am talking my head off through here, I kind of get in my head, I start talking, I start teaching. If y'all don't stop me, I'm just assuming that y'all get it, right? Like, okay, all that makes sense. Let's move on. Let's not waste time. Um, so if you need me to slow down, if you need me to go back, hey, I missed that. Hey, I was writing down this other thing and I missed what you just said, right? Whatever it is, then as long as you're not just, watching TikTok videos and then you want me to go back and reteach something. Of course, that won't make me very happy. But whatever it is, please don't be afraid to stop me and interrupt me, right? I may stumble over my tongue for a second as my brain tries to catch up with what you're asking. And I may not hear you the first time just because my brain is a little bit like a thunderstorm all the time. And I have to really focus on people to hear and understand what they're saying. But that doesn't mean I don't want you to interrupt me, right? Y'all are paying for this. Somebody's paying for this, even if you're not, right? I'm being paid to be here, so I want to give you what you need, right? This is not a theater show where I'm going to be up here and I'm going to do my thing and you can either enjoy it or not, right? This is interactive. So feel free to ask me questions, talk at me, let me know what you need, even in the middle of class. We are typically getting done with plenty of time left over, right? That extra time is designed for some of the back and forth. Now, if nobody's ever asking any questions, I can do things like make quiz games that we can pause and play throughout the lecture. All right. I don't. That feels good and it can be reinforcing. Um, and so, we can do some of that. That's fine. But ultimately, I don't know that it has a ton of value. Um, One of the things I've talked to a couple of students over the past few days that we've been here, and particularly after that Friday study session that we had this week, and there were some things that I thought were notable, right? Some people dealing with different learning disabilities or just completely new to the topic. And I got the impression through the conversations that I was having that they were worried about getting through all the material in a short amount of time more than they were worried about understanding the material, right? Yes, there is a lot that we have to cover. There is a lot to keep up with but for every exam. However, right, if you don't understand what you're reading and you don't understand what you're studying and it's just a big long list of words, right? Well, there's two big problems with that. And the first one is it's really hard to remember a whole bunch of words without any context, without anything to hang them on, right? In education, we call it scaffolding. Like you think about it like a closet in your house, right? You don't hang your clothes up on the drywall. You got to build the shelf and put a curtain rod in before you can hang your clothes up in there. So, um, we really need to focus on connecting the pieces together, and adding all of that. Now, the human brain, right? It's kind of weird. Vocabulary words all on their own without context, you can memorize them, right? But it's actually easier to remember something and recall it in life when you're in front of a patient and on a test when you have to produce an answer when you have 12 or 15 different things all connected together in your head, right? Like, what is simple squamous epithelial tissue? Okay. Well, you could just have a note card and say, okay, well, it's a lining that's made of a single layer of flat cells that look like scales, textbook definition. Or you could have that definition and you could also have in your head, these are the 50 pictures I've looked at it like this, and we find it in the kidney, and this is where we find it in the kidney, and this is what it looks like, and this is where we find it in the inside of blood vessels, and this is what it looks like, right? And it's really thin, and that makes it really easy for oxygen and nutrients and fluids to pass through it, right? And we can put all this information together. We build the context, all of that stuff together. You're like, oh, and you begin to make a pattern out of it. Well, where do we find simple squamous epithelial tissue? We find it in areas where we need gases, dissolved gases and things to be able to pass through, right? Like the air sacs of the lungs, like the glomerulus of the kidney for filtration, right? So diffusion and filtration is the category of functions. And then all of that kind of starts to fall in place together and you understand, and it's not so much, let's just memorize this list of facts. Okay, so I'm going to do my best to try to emphasize the interconnectedness of all things, if you will permit me to plagiarize one of my favorite authors with that phrase. But I think that is helpful on how to study. So, practically speaking, you're like, okay, great. Practically speaking, what I would encourage you to do is slow down, spend more time up front gathering all of the pieces of information together and understanding the whole picture, right? And then not only you will remember more of it the first time, but you'll be able to tie it to the other stuff down the line, and it'll be easier to remember. It'll be easier to bring up out of your head during a testing situation and on into the future. And that's how you learn things forever and don't forget them, as opposed to sort of cramming everything into your head on a test and then, forgetting 70 or 80% of it over the next couple of weeks because you never use it again. So, all right. That is the end of my sort of encouragement session there. And if you want to come meet with me and just talk about study strategies and different tools, that's fine. We also have tutoring available, less available in the evenings and on Fridays, unfortunately. But we could hook you up with, I think they're called success coaches on this campus, to help you plug you into whatever resources you need. And I know a lot of the tutoring is available in the evenings as well. So, um, sure. Now, I will say I've gotten some feedback this week. I had some people go to the tutors, and some of them were like, yeah, it was great. And then some people were like, I asked a question and then they talked about stuff I already knew instead of answering my question. So tutors vary. Your mileage may vary. There we go. So we'll go ahead and get into tonight's topic. I do want to try to at least introduce protein synthesis to you, even though we're probably not going to be able to finish lecture number 7 tonight, and you will need to watch the pre-recorded lecture video in Blackboard by Dr. Foskett. You find that it'll be under the Unit 1 materials, right? Lecture videos, and it's the very last link on that lecture, on the lecture videos, or Unit 1 lecture materials, rather. Yes, ma'am. Mhm. Okay, sure. So her lecture videos, I think were recorded maybe 5 or 6 years ago. And so we're always updating the materials and changing things out, but the core concepts are still there. But as always, if there's something on our PowerPoint slides that she doesn't cover and you need it explained, shoot me a message on Blackboard. We can meet during office hours, or if I'm getting the same question from a lot of people, I'm happy to even record a video and post that for you, just as an addendum to kind of cover whatever gaps there are in those lecture videos. So, um, yeah, I'm more than happy to do that. Also remember that we have the supplemental videos that are sort of like little short, more entertaining, more digestible videos from YouTube that will cover individual topics briefly, instead of getting involved in a 2-hour lecture video that you need to play on 1.25 to get through, right? You can digest an individual topic and at least get the basics of it in 10 to 15 minutes in most of those videos. So, all right. So tonight we're going to talk about the cell, the contents of the cell, and the organelles and their functions, right? So we're going to be talking about sort of a generalized animal cell, and um, I'll say, depending on when you went through high school, right, your experience in freshman biology may be widely different. When I went to high school, we spent like 8 weeks doing animal and plant cells and all the parts, and we really got into it and did a whole lot of experience. My daughter, who took freshman biology about 3 years ago, she's a senior now, they spent about 2 weeks on the cell, and then they moved on to, conservation and ecology and climate change, which, all fine topics, good to learn about, but Why in the hell are we discussing that in biology class, right? The cell is the big thing. So, some of this will probably be review for you. Some of it may be brand new. I think everybody remembers, what is the mitochondria? The powerhouse of the cell, right? We could all chant that together. Um, but we'll try to just tease out some of those details here. So, what is inside the cell membrane, right? The cytoplasm. Now, when I was in high school, it took me a while to get this distinction, right? The cytoplasm is the watery component, and sometimes it's more a gel because there's so much stuff dissolved in it. And then also all of the organelles and all of the junk that's floating in it, right? So the cytosol is the water with all the dissolved proteins, salts, and sugars. The cytoplasm includes all the organelles, all of the proteins and pigments and vesicles and so on and so forth that are floating around inside of there. So you may be asked at some point, even if not on our lecture exam, to distinguish between cytoplasm and cytosol. That's the key difference. Now, within the plasma lemma or the plasma membrane that surrounds the entire cell, of course, the organelles are floating within that cytoplasm. Some of these organelles are also membrane bound. They're membranous, meaning that, there's an outer membrane. The mitochondria is such an example. In fact, the mitochondria, let me not step on my feet. We'll get to the slide and I'll say it then. But the mitochondria actually has two membranes, right? An outer and an inner membrane. Peroxisomes and lysosomes are basically just like big membranous storage tanks, big bubbles that hold on to certain enzymes and chemicals for digesting and or detoxifying substances, or in some cases, initiating the process of apoptosis or programmed cell death if the cell detects that its DNA is damaged beyond repair or it's infected with a virus or something. The cell may decide to destroy itself rather than continue to be infected, right? The endoplasmic reticulum, right, is a set of membranes and channels surrounding and connected to the nucleus that functions both as a manufacturing site as well as a transport system. And then finally, the Golgi apparatus is a second little set of membranes that functions as a bit of a post office, particularly for proteins designated to be secreted out of the cell, right? So if our cell is making a protein that it's going to expel, bless you, right? The Golgi apparatus is going to modify it and pack it up in a nice little bubble and get it ready for shipment, if you will. Now, some other organelles, some other structures are the cytoskeleton. It's made up of some protein fibers and filaments and tubules, right? Things like actin filaments, microfilaments, intermediate filaments, and microtubules. The centrioles we looked at in lab for mitosis, and then ribosomes, which are an advanced combination of protein and RNA made up of two subunits that work together to actually translate other RNA to create proteins. So they're like little manufacturing robots made up of those things. And when you cover protein synthesis, you'll see the RNA and protein component of the ribosomes. That's going to be covered in lecture number 7. So now we have the membranes again, because membranes separate things, right? That's the big thing. So mitochondria, the powerhouse of the cell. So we have an outer membrane, and then we have some inner membranes, which sort of appear as squiggly lines, and that's how they're usually pictured in our artistic drawings in our textbook. The mitochondria takes the products of respiration, like glycolysis and the citric acid cycle, and then moves them through what's called the electron transport chain, which oxidizes the food, essentially, that we feed into the system in order to create a lot of ATP, right? And so they require the presence of oxygen. Now, an interesting fact about mitochondria is that they appear to be, they appear to have an origin as an independent life form, an independent bacterium, that merged with animal cells at some point and formed a symbiotic relationship where they got fed and then they created ATP for the rest of the cell, right, in exchange for being taken care of. And so over time, they are heritable, right? And the mitochondria can divide and can replicate. So under the right circumstances, you can actually make more mitochondria, and that's generally considered to be a healthy thing because you can then your cells can make more energy, right? And under the wrong circumstances, right, your mitochondria can become thick and begin to diminish in number, right? Mitochondrial DNA is distinct from the DNA that makes up you, right? And it's actually passed down strictly from your maternal line, right? So it's from your female ancestors. And so if we want a very accurate maternal lineage, right, we can track the changes in the mitochondrial DNA over time, right? So here's what they look like. So we have our little diagram there. You can see the two membranes. The folds are called cristae. And then over there on the right, marked C, that is an electron micrograph that has been colorized of an actual mitochondrion. So we can see the intermembranous structures there. And of course, the enzymes involved in the electron transport chain are embedded within that inner membrane. Now, ribosomes, I kind of previewed this for you. This is where proteins are made, right? From the simplest dipeptides, which are two amino acids joined together, to the most complex proteins like enzymes and things like hemoglobin, right? Ribosomes are where it happens. So most of the ribosomes we're going to deal with are going to be bound in the rough endoplasmic reticulum, but there are free-floating ribosomes within the cytosol that can manufacture proteins. Mostly the free-floating ribosomes are going to be manufacturing proteins that are going to stay within the cell, and inside the rough endoplasmic reticulum, we're going to be dealing with proteins that are going to be exported or bound into membranes. And so that brings us in, well, if the ribosomes are embedded in the rough ER, then what in the world is the endoplasmic reticulum, right? So again, it's just a bunch of interconnected tubes and cisterns, right? Parallel membranes forming these pockets and channels where different substances are synthesized or modified, so on. So the first one, the rough endoplasmic reticulum, is called rough because you see the little ribosomes being embedded into the membrane, and it's sort of like putting a whole bunch of buttons or a whole bunch of sequins on a shirt, right? You can see the texture there, so that makes it rough. The smooth endoplasmic reticulum, and rough deals primarily with proteins, right? Protein synthesis there. And then the smooth endoplasmic reticulum has enzymes embedded within the membrane, but they're not so large and visible. And this deals primarily with like lipid metabolism. So if we're breaking down fats inside the cell or manufacturing lipid substances, that's happening in the smooth endoplasmic reticulum. And it also deals with the transport of those substances to different places in the cell. In addition, right, certain drugs and pesticides are fat soluble, so detoxification happens here in the smooth endoplasmic reticulum. So a lot of toxins are non-polar, meaning they're fat soluble, just like oils. And so in the liver in particular, when we're detoxifying things, there's several different protein pathways that work to break down toxic substances. But essentially what we're trying to do is we're trying to modify the larger molecule and break it up into pieces that will be polar and therefore water soluble, right? And so the idea is if we can break it apart and make it able to be dissolved in water, then we can pass it on to our urine and get it excreted out of our body very easily. So, sometimes that works very well, sometimes not with great effects. Alcohol is a non-polar substance. So We have ethanol, which is the kind of alcohol that some of us enjoy in things like wine and beer and other harder beverages, right? Methanol is a very similar alcohol, but it's derived from the fermentation of wood, if you will, and cellulose. Now, they're very chemically similar, a lot like the different sugars that we looked at, right? They have a very similar chemical formula, but ethanol, we detoxify, right? It's technically a poison, but we can detoxify it, we can use it for energy, and fairly rapidly pass it out of our system in our urine, and some of it actually gets passed out through our lungs. Um, but methanol, as it's being processed in the liver, there's an intermediate step that turns it into formaldehyde, right? And so methanol is very, very poisonous. So thinking back historically during Prohibition, bootleggers would often adulterate their moonshine with methanol, right, to stretch it out, right, for the same reason that all drug dealers from every place, anytime, add other junk to the product, right, to be able to sell more of it. And so there are stories from that era of people drinking moonshine and then going blind or dying, right? It turns out formaldehyde kills the eyeballs pretty quickly, and it is not compatible with life. How do we know that? Well, we use it to float dead things in so they never rot, right? That's that preservative solution. Um, so yeah, so practically speaking, unless you have a toddler with access to IndyCar fuel at the house, you're probably never going to see this. But if somebody does come down with methanol poisoning, because they've consumed methanol in some form or fashion, the treatment is actually to give them ethanol, right? We give them IV, basically, what's the name? Pure grain alcohol, Everclear, right? We add ethanol to the bloodstream, not because it's good to get them drunk, but because ethanol and methanol compete for the same detoxification pathway. And by adding ethanol, we can slow down the processing of methanol. That way the formaldehyde levels don't build up as high or as quickly. I've already told you the story. I'm getting looks like I've already said this. Okay, cool. All right, so that's just an interesting little aside there. So that's the detoxification here. Glycogen in muscle and liver cells. We store glucose, right? And we can build long chains of glucose. That's called glycogen. It's like the starch that plants produce, right? But we're using glucose and not fructose, and that branches a little bit differently. We don't need to get into all that. But so when we store that energy inside the cell, the liver cell, for instance, if our blood sugar gets low, if we haven't eaten in 12 or 16 hours, then we have a hormone called glucagon that stimulates glycogen breakdown in the liver, and then the liver cells will actually release glucose back into the bloodstream to bring our blood sugar levels back up. And muscle cells can also store a small amount of glycogen as well. And then, of course, storage and release of calcium there. So here's what this looks like in the picture, right? The endoplasmic reticulum is directly attached to the outer nuclear membrane. So the nucleus, we just found out, right, is another membrane-bound organelle within the cell, and it has two membranes, a double layer there, with pores in it. And so a preview of protein synthesis, a bunch of stuff happens inside the nucleus, and we end up with a strand of RNA that we call mRNA, messenger RNA, and it leaves those little nuclear pores and will drift into the rough endoplasmic reticulum, and then the ribosome will grab hold of it and start decoding it and plugging amino acids in to make the protein that that particular thing codes for. So most of what we see in this picture is going to be roughened ER. The little black sand sort of embedded in the membrane there, that is representing a lot of the ribosomes. As we get up here, just to the very top left corner of this picture, we can start to see more of the smooth ER here. So, right, there's not a lot of it visible here. Most of this is rough ER, but, you know, it is there. Now the Golgi. Again, you can think of the Golgi as like the post office. This is the shipping, packing department of the cell, and it is a lot of flattened membranous sacs. It looks a little bit like the endoplasmic reticulum, similar in structure. And so when proteins get released from the endoplasmic reticulum, they'll be packaged up inside of a little vesicle, like a little bubble of membrane, and that's going to bind to the cis face of the Golgi apparatus. So what does cis mean in this context? It means the same side. So this ends up being the side of the Golgi that is closest to the nucleus. So basically the little bubble comes out of the ER and then just goes right into the nearest face of the Golgi apparatus there, right? And now within the Golgi, the proteins may be modified, they may be repackaged, and they're transported across the Golgi to the face that is closest to the plasma membrane, the outer surface called the transface. Trans just means across, right? And so then the completed protein will be put inside another little vesicle, another little bubble, and be shipped out to its destination. So an example, in our nerve cells, we make a neurotransmitter, right? It's a protein. So in motor neurons that we're going to be looking at very soon, we make a neurotransmitter called acetylcholine. And so we make that up, and glutamate, epinephrine, right? All of these are examples. But we make this little protein and we put it inside of a little vesicle, and that's all happening in that cell body, right? The soma. And then that little vesicle gets transported all the way down the axon, and it gets stored right at the axon terminals, the tiny little branches right at the very end. And it just sits there and it waits, right? We wait for one of those action potentials. Right? What a depolarization of that membrane. And when calcium channels open at the very end of that whole process, calcium becomes a signal for those little vesicles to bond to the cell membrane and release their contents to the outside space, the extracellular space. Something really grabbed me. So that's an example of a protein being manufactured and then transported and destined for exocytosis, right? Which is the active transport involving the changing shape of the membrane, bonding a vesicle to the interface and opening its contents up to the extracellular space here. Now, that is number one here, right? On this particular list, 3 types of vesicles that are going to bud from the shipping face, the transface, which is the face that is furthest away from the nucleus and facing the outer cell membrane. Right. The second type, we have little vesicles full of lipids, or made of a membrane that has lipids and transmembrane proteins bound into it. And so these vesicles may be joined with the cell membrane, or they may be joined with other membranous organelles. And so if we're modifying the organelles or repairing different organelles, that may be happening there. And then the third would be like the big storage tank that's just going to wait around in the cell until it's needed, like a lysosome or peroxisome. And it never leaves. So here's one of our little diagrams here that has all the bubbles. And so in step 1, protein containing vesicles pinch off the rough ER and migrate to fuse with the membranes of the Golgi, right? We could add a little caret in there and say the cis face of the Golgi apparatus, right? Now within the Golgi, right, proteins are modified within the Golgi compartments. Very bland, right? If I was trying to memorize that, I'd probably forget it too, right? But things are being changed and repackaged, right? If you want to think about it like the post office, or perhaps more aptly, the shipping department of a manufacturing warehouse, right? Where they're taking product, they're packaging it up, wrapping it up, putting it in the right size box, and then stacking it on pallets And then those pallets are loaded into a pallet rack and then eventually loaded onto a truck, right, or a train or wherever. That's the Golgi apparatus there. So pathway A, that's exocytosis. That's the neurotransmitter I talked you through. Pathway B, right, there's not really anything important within the vesicle. What's important are the lipids and cholesterol and the transmembrane proteins and glycoproteins and glycolipids that make up the membrane that is the vesicle, and then that just gets added to either the cell membrane or to another organelle. And then pathway C, right? We've made a big old tank of nasty stuff that it's very useful, but we need to keep it away from everything else so we don't destroy the whole cell, right? So in certain white blood cells or in osteoclasts, those cells that dissolve bone, they have a lot of these types of chemicals, acids and enzymes and so on and so forth that can digest um, bone in the case of an osteoclast, or in the case of certain white blood cells like macrophages, they'll fuse the bacteria they engulf with a lysosome or a phagosome to digest the bacterium and destroy it, and eventually present the different parts to other white blood cells so they can start making antibodies. So peroxisomes are in this class, right? Contain powerful oxidases. We can see that oxy in the word there, and catalases. And so again, oxidizing is one way that we can break things down and sometimes make them more polar to be able to be excreted. And so most of us have used hydrogen peroxide at some point, H2O2. And if you spray it on a superficial wound, very often we'll see sort of an aggressive bubbling or foaming. Um, that is the H2O being converted to H2O2 being converted to water and oxygen by an enzyme in our cell called peroxidase. Right? Peroxide is actually very, very harmful to cells. It's very powerful. And so we have defensive enzymes against that very chemical. Um, that bubbling action can promote the release of embedded dirt and things up out of a wound, so it can be somewhat helpful. But with deep wounds, don't use peroxide. It's too aggressive, right? If it's more than just a superficial wound there. So there we go. Lysosomes, right? To lysis, to cut. So these are typically things if we're needing to break things apart. The enzymes will be in a lysosome, but similar idea, right? We're chemically attacking whatever gets fused with it there. And then now we're going to finish off this little section with the cytoskeleton, right? This is kind of a little many category within all of the different organelles because it contains a lot of different parts. The three major ones are microfilaments, intermediate filaments, and microtubules. And some of these proteins we're going to see later in the semester as well, like this one, right? The microfilaments are made of the protein actin. Actin. These actin can be contractile, right? Meaning it can move and shorten. And we'll see this again when we discuss our skeletal muscle tissue at the microscopic level, right? So on the inside of the cell membrane, right, we have a web of these proteins attached to transmembrane integral proteins and peripheral proteins, making this web just inside the cell, right? And so it's these proteins that allow the cell to use energy to change its shape by contracting and moving because it is bound to the inside of the membrane itself. Now, a little bit larger, we have intermediate filaments. These are wound together like a rope, actually. Um, when we looked at desmosomes and we had those filaments coming through the desmosomes and kind of hooking up with the other cell, right? Those were intermediate filaments there. And then in places like our skin, right? Keratin filaments in our epithelial cells are intermediate filaments there. right? And then finally, we have microtubules. These are hollow tubes made up of spiraling subunits of proteins called tubulins. And these have a much larger, since they're so large, they contribute more to the shape of the cell. And if organelles or other substances are being transported around the cell, there are actually proteins that look like little legs, essentially, that just walk down a microtubule while they're attached to larger organelles or vesicles or even just molecules and to move them around to different parts of the cell. That's pretty cool. I'm going to pause that for a second and see if I can bring up a quick animation. No, I don't have the YouTube Premium on this computer, so we got to watch ads. Skip. And give me full screen. So we see here a kinesin motor protein, right? It looks almost like an old cartoon character, right? But it's walking down the microtubule there. Let's replay that again. Walking down the microtubule, transporting a vesicle to different parts of the cell. So yeah, it's pretty interesting, right? So this is an active process, right? So it does require energy, but pretty cool stuff going on there. That's sort of a short video. There are examples of longer format videos narrating that. Let me see if I can find an actual electron micrograph. There's another 3D animation. This is another 3D animation of the same thing. YouTube probably isn't the place to look for a picture, is it? Image search. Here we go. So here are some examples. Right, and so when we're using an electron microscope, um, the tissue has to be preserved and fixed, and so we get different views by looking at different things. We can't look at living cells like this. The bombardment by the electrons would kill it. But let's see if this video, click on the link, is any better. Again, I don't know that we need to show the, or have that narration. Stop talking, we're just here for the pictures. Meh, 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 meh. There we go. So this would be an example of, looks like they're using a fluorescent sort of technique to highlight some of these things. And this is several different pictures combined together to show range of movement of another protein called dynein. And then taking a look, we've molecularly modeled some of these proteins. So this is an image of what the protein would look like in its three-dimensional folded shape there. So right. All that information is out there if you want it. get back to this. All right, now centrosome and centrioles, right? The centrosome is like the quote unquote center of the cell. Things are anchored to this point. And we saw that when we were looking at mitosis in the lab, right? When the centrosomes separated, right, and migrated to opposite poles of the cell, we were essentially moving the central cell anchor to opposite ends in preparation to use it to yank things apart, right, as those chromatids separated in anaphase there. Centrioles are really interesting structures. They're little barrels, essentially formed of three microtubule subunits. And not only will you see this in the centrosome rather, you'll see it anywhere you see like flagella and cilia attached to the cell membrane. These centrioles are going to make up part of that base that binds that structure to the cell membrane and is part of the little molecular motor that burns ATP and actually causes them to spin and move around. So we've taken a look at an example of cilia in our pseudostratified ciliated columnar epithelium in our last unit. Of course, right, they beat rhythmically in a circular motion, and the air passageways to move mucus and debris up and out of the air passageways, right, they also exist in other places. The tail of a sperm cell, a mature sperm, right, it swims through the use of a flagella, right? Works in much the same way. It spirals around in a little circle and whips back and forth to propel the cell down. And there are bacteria that use these same structures to move around their environment as well. Microvilli, we looked at those in our simple columnar epithelial tissue that we find in the digestive tract, among other places, as well as, I think, in the uterus. A few other examples. But zooming in on them, right, they're little finger-like extensions. This is cell membrane, right? They're still filled with cytosol and cytoplasm, increasing the surface area for absorption of nutrients. But this picture is cool because we can see the actin filaments inside them binding them to that inner terminal web. Right, so we zoomed in enough that we can actually see the cytoskeleton there. That's pretty neat, right? Now, finally, the nucleus. Large, generally the largest organelle. It contains the DNA, right? Our genetic code, the stuff we need to make all the bits and pieces that become us when we put them all together here. There's a lot of really complex signaling that goes on inside the cell. And the DNA works, or the nucleus rather, works more like the brain of the cell, responding to signaling molecules to make decisions about what proteins need to be created at any given time, right? And so DNA, like we learned in lab, is a double helix structure, right? And then we wind that double helix around these little pill-shaped proteins called histones, right? 8 of them stacked up together. And that's how DNA is sort of stored most of the time inside the nucleus. And we looked at how that coils up even tighter into that chromosomal form as we enter the mitotic phase, right? But the DNA functionally, when we need to use it inside of a normal cell that's just going through interphase, we unwind it from the histone proteins, and then we take the 2 strands and we sort of unzip them at the location that codes for whatever protein we need to create, right? And you'll get into how that works in lecture number 7 in more detail. But then in the process, right, we read the DNA and create a new molecule of RNA, a complementary strand of RNA called messenger RNA, right? Now, back in high school, you probably learned, all right, we create the mRNA, and then the mRNA leaves through the nuclear pore, and then it becomes a protein. That's broadly correct, but there's an intermediate step there. The raw mRNA that gets created, right, in the process of transcription, then is edited within, like there's some dead space within the code that's sort of cut out and cleaned out, and then it leaves, and then it becomes a protein. So in the protein synthesis lecture, you're going to talk about transcription and translation. Transcription's a whole thing, translation's a whole other thing. So let me just talk to you for a second about what those words mean. Now, again, I'm going to go back way back in history, way back before we had the Gutenberg printing press, right? People made copies of the written word by just making copies of the written word. Like there was a job called a scribe where that person was essentially paid and their whole job was to read things and make accurate copies of it, right? Not, back then it was very common. Not everybody could read, not everybody could write. And so to transcribe is to copy across, right? So to take your original work and then to copy it across. And so typically also in the same language, right? So if I have a German original work and I'm making a copy of it, the copy is also going to be in German. Cool. So in transcription inside the cell, it works the same way, right? The original work is the DNA, right? And then to scribe it, we keep it in the form of a chain of nucleic acids, right? DNA to RNA, it's still nucleic acids, it's still the same language, right? But maybe we took the great big museum piece Bible, right? That's like 4 feet tall and 3 feet wide, and we copied it into a smaller piece that could be carried by hand around, right? So the DNA, huge thing that we can't really take out of the nucleus, but we copy it into something that's more portable. That's transcription, right? So from nucleic acid in the form of DNA to nucleic acid in the form of mRNA, right? It's the same language, different format, right? Maybe the handwriting's different. Maybe we leave out all the fancy drawings and the big first letters at the beginning of sentences, but it's the same language. Now, after the mRNA gets pushed out of the nucleus, and it enters the ribosome and we start making proteins, that's translation, right? We're changing the language. We're taking German and we're turning it into English, if you will, right? That's just easy. It could be any language, right? That's not the point. But we're taking a chain of nucleic acids, right? And we're turning it into a chain of amino acids. We're going from genetic material to protein. It's an entirely different class of organic compound, right? So it is likened unto translating from one language to another, right? Translating from French to German or whatever, right? Whatever 2 languages you want to use in your example. So all of that is going on inside the nucleus. Now also within the nucleus, we have the nucleolus that is densely packed RNA, and there's a few different types of RNA. And some of what we call the ribosomal RNA gets manufactured in that region. So here's our diagram. So again, the nucleus has 2 membranes. It's a double layered membrane. And those 2 membranes are bound together with gap junctions. I hope you recognize that particular shape with the little flower petals and the little tunnel in between them there. And again, it is that outer membrane is directly connected with the endoplasmic reticulum. So those pores, they're going to exit right into that. So, yeah, okay. Now the other detail there is the little holes in the nuclear membrane. They're very small, right? That's why the DNA can't get out. A lot of things can't get in. So yeah, that regulates things in there. Yep, I've already said that. Good to go there. And we've already talked about this, right? The chromatin form, as we see, DNA wrapped around those big aspirin pill looking things. And it turns out that makes up about 60% of the bulk of chromatin. There's some RNA involved in there too. We looked at that when we did mitosis, but here it is visually again. I think we saw this picture in lab as well. So since it's a process, since we have the little circles with the numbers on it, we'll go through this. So our raw DNA, right, is this double helix format. bound in the middle. The complementary nucleotides are bound with hydrogen bonds, right? And then that wraps up around the histone proteins. So there's 8 histone proteins wrapped with a double wind of the DNA double helix. And so if we use an electron microscope again, right, it looks like beads on a string like that. I think that's pretty cool. Strand of Christmas lights if you want. And then as we condense down more into the chromosome, right, it condenses and spirals in on itself and forms a more complex spiral shape there. So, yeah, fun times. Right. Now, cell cycle, we talked about it in lab. We went into a lot of detail. We went through everything bit by bit. I spent like 40 minutes on it. I'm not reading this to you again, right? A detail that I noticed, not just in this class, but across all the classes for the lab exam, the S phase is not the synthesis phase, it is the synthetic phase, okay? Synthesis is what happens in the synthetic phase, but the name of the phase is the synthetic phase, right? S phase is the synthetic phase. Most everybody correctly got that question right? They identified it was the S phase, all of that fun stuff. But that was a little wrinkle that some folks lost partial credit on for spelling. So these slides are exactly the same as the ones we already looked at in labs. So again, I'm not going to waste your time going through that again, unless somebody has a specific question about any of that content they'd like me to review. Hearing only crickets, I'm going to move on to the next thing. Phenomena. All right, now seems like a good time to take 5 minutes. We'll come back in here. I'll start and hit the high points of some of the protein synthesis stuff, and then we'll break and move over to lab. What I did to this arm. Feels like I was doing roll-ups at the gym with this arm today. I don't remember doing any of that.
Yes. Sorry. She so last 3 days I got the yeah, on the quiz, I don't know how it's going. And yeah, it's for a 4 period, just exactly the number of it's not exactly. It's no long. I was not 13, 14. Yeah. So that is one of the great one the first day I didn't come because it was like really bad. Look, so 4 days for those. Yes, 4 drops. I do drop in some. 1, 2, 3, 4, out of 4. And 440 out of I got 38. I was thinking like maybe they were more she's a member. Maybe this one is together. Yeah. I'm like, try it down. Not, I hate it on here. When you get the week of yes, it's 26. But we have already done this before. All of the ones that can find here. Yeah, that's right. Only the ones is here. I don't know that. I just found everything. So the homework is, I will read it back to school. Since it has like 7 minutes. If we need the budget all the homework we already do. It's one. Is Mr. We have lab today?
Yes, we sure do. Yeah, okay. All right, so this lecture again, you really do need to watch the complete lecture video on Blackboard to get all of the details, but just kind of want to get the high points, kind of start, building some coat racks for you, if you will. So when you jump into it, you'll have, something to connect the information to. All right, so DNA replication, of course, is just making a copy, right, of the DNA. Y'all know already that this happens during the synthetic phase of interphase. So we begin to look at how that works. Now, we don't unzip the entire DNA molecule all at once and then make a copy. We have what's called a replication bubble, where a small portion of a DNA strand will be unzipped, and then a complementary strand is added along the length of the DNA strand, and the replication bubble moves as those complementary nucleotides are being added. So the enzyme that is going to add the nucleotides to the new strand of DNA is DNA polymerase, right? A polymer is just a chain of small repeating subunits, right? DNA and RNA are polymers, right? So DNA polymerase is creating a new chain of nucleotides and turning it into DNA, right? That's how that name goes. Now, DNA polymerase is going to work in one direction, right? An enzyme is designed to read DNA from one end and go to the other. But DNA in its double helix arrangement, because they are complementary strands, right? They are oriented in opposite directions, right? We have top to bottom on one strand and then bottom to top. Or yeah, it's flipped over on the complementary strand. And so the terminology we use for the different halves of this process going on at the same time would be the leading and the lagging strand. So the leading strand, as it's unzipping, DNA polymerase is just going to be able to travel down that long chain of DNA, that single strand, and it's going to continuously add new nucleotides as the bubble moves, right? But the bubble is moving in one direction. And so on the lagging strand, the DNA polymerase is going to read a short segment, detach, the bubble's going to move, it's going to move over, read a short segment, and it's essentially reading in the opposite direction, right? If it's reading from right to left, the 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 unzipping is going from left to right. So it basically has to catch each little chunk as it can as the bubble moves from left to right, making little fragments of DNA that we then bind together to create the whole piece on the lagging side. The little pieces of DNA that get created there are called Okazaki fragments. Hmm? Yes. They're Okazaki fragments. Yep. So the little pieces of DNA that are created on the lagging strand are Okazaki fragments. That word discontinuously means it's not just making one long strand, right? It's making a little piece, breaking off, making a little piece. So it's broken up. It's not continuous. And then the enzyme there is DNA ligase. Now, good news for enzyme names, right? They actually mean something. So once you learn like a base enzyme word, then we just add the descriptors in front of it, right? So DNA polymerase, the polymerase is anything that makes a long chain of pieces, right? DNA polymerase specifically is making DNA from a long chain of nucleotides. RNA polymerase would be making RNA. Ligase, a ligand is a binder, right? It's a connection. So DNA ligase, that just means it's an enzyme that's binding pieces of DNA together. And that's what connects the Okazaki fragments. Let me see if I can get this camera to follow me around. As long as I fix it and move slowly. There we go. It's not recording me anyway, I'm just playing. All right. So at the end of this replication process, what we'll end up with is two identical DNA strands, right? We've made a copy, but each strand is made up of one strand of the original copy of the DNA and another new strand, right? So half of it will be old and half of it will be new. The, the term we use for that arrangement is called semi-conservative replication, right? So let's break that term down. Replication is copying, right? Semi is half, and then to conserve is to save something, right? To preserve something. So semi-conservative, we're preserving half of the original, and then we're making a copy, right? Semi-conservative replication. So, and each new DNA molecule right? Each one of those has one strand of the old and one strand of the new. So when we undergo mitosis, one of those copies will end up in one daughter cell and the other one will end up in the other daughter cell. So each cell gets a complete copy of the entire double helix of the DNA here. So in this picture right here, we can see that this replication bubble is unzipping from right to left. So our leading strand is on top, right? And that replication fork that we see here right? Right here, this is where the DNA is being unzipped. And that is being performed by an enzyme that's doing that job to expose those bases. It's separating the hydrogen bonds in the middle of this double strand. Now our DNA polymerase here is reading the old parental strand here, right? And then plugging in the complementary nucleotides that match up as it goes. And since we're unzipping from right to left, and this DNA polymerase is reading from right to left, it can just sit there on that DNA strand the whole time. It never has to separate and rejoin. Now, the lagging strand, remember, each strand of DNA is oriented in opposite directions. And so this DNA polymerase down here is reading from left to right and adding complementary nucleotides from left to right. Right? So it has joined right here at the front edge of our replication fork, and it's moving this way, right? But then we're going to continue unzipping in this direction, and so we're going to have another DNA polymerase plug in on the left and read to the right until it gets to the old piece, or to the piece that's been created. And we're continuously having to plug in a DNA polymerase and read those short chains, right? That's how we end up with the Okazaki fragments there. So, yeah. So that's DNA replication. Now, that's important to understand because when we are doing protein synthesis inside of a cell, we're using some of these same processes to open up a replication bubble, right? But instead of using DNA polymerase to make a copy of DNA, we're going to use other enzymes to make RNA, right? That's that transcription I mentioned to you a little bit ago. So all DNA is a master code for the proteins that make up the pieces of your body. So each gene then is a segment of DNA that codes for a single polypeptide or protein. Now, we have the bases, right? The nucleotides. And we read them in sets of three or in triplets. So each three base sequence in order is coded for a particular amino acid, right? And when we make that, we can read that there. So in DNA, the letters for the bases are A, G, T, and C. Adenine, guanine, tyrosine. So thymine, that's the one. Yep. Thank you. And cytosol, something like that. I don't know. We'll see it here in a second. That is not not coming to me right now. Right. So anyway, the complementary pairs, A and T match up together and G and C match up together in DNA. Now, in RNA, we have a different nucleotide, a different base called uracil that's substituted for thymine. So instead of AT, if we're reading DNA to create an RNA, then we're going to have AU and UA, right? So there we go. So Now, as we're reading the DNA, right, I told you there's some junk stuff in there from the mRNA that has to get filtered out. That's because not every sequence of nucleotides in our DNA is actually meaningful information. We have some spaces in between genes that just sort of separate them. And we have a lot of long chains of sort of what we might call junk DNA that doesn't really code for proteins, but it's embedded. Sometimes, we can even look at our DNA and see where viral DNA has been spliced in different places. But the parts that actually code for a protein are called exons, right? And then all of the filler, if you will, all of sort of the administrative stuff, like in a library, the books would be the exons, right? And then all the bookshelves and all the bookends and all the things that separate the books, right, would be the introns, right? They're necessary, like we need them for structure to keep things organized, right? But that's not where the good stuff is, if that makes sense. Um, so when we create a strand of mRNA in advance of creating a new protein, right, we have to then process that mRNA or messenger RNA to remove those introns from that code, because if we left them in, then we'd end up with some kind of nonsense, non-functional protein here. So that's mRNA. Ribosomal RNA makes up one of the subunits. It's part of one of the subunits of the ribosome. And we need that in the ribosome because it contains some nucleotide bases that are going to be there, right? The structures that are going to match up with the mRNA and be able to help decode it, right? And then transfer RNA is a special type of RNA that actually connects to the specific amino acid, and then it has an anticodon, which is complementary to each codon as it goes in there. So we'll see how that process works there. So transcription and translation, I sort of previewed that for you. This is it, right? DNA to our pre-mRNA that still has all that intron nonsense in it, and then we process it to form our final mRNA. mRNA is going to exit the nucleus via one of the nuclear pores and then find a ribosome. Maybe it's in the rough endoplasmic reticulum, maybe it's free floating in the cytosol. Doesn't matter, but the ribosome does the job of translation, right? Reading the original language and taking it into the new language. So, in order to create a copy of mRNA to begin to create a protein, we have to do the same sort of sequence, right? We have to unwind the DNA from the histone proteins, right? We need to bind to a specific sequence that indicates the beginning of the gene code for that specific protein. We need to unzip, and then we need to bind the enzyme that's actually going to create the mRNA, right? So there's a lot of little steps involved in getting this started. Broadly speaking, these little activators are called transcription factors, right? What do we need to do to start to make a copy of this book, right? We got to get in the room, we got to have our ink, we got to have our paper, we got to have a place to write, we got to have enough light to see. All of that stuff might be the transcription factors here. So RNA polymerase, right? We already talked about DNA polymerase, right? Making a chain of DNA. RNA polymerase is doing the same thing, right? But it's making RNA. So again, the names actually are meaningful, which is helpful. So the RNA polymerase is going to separate or unzip the two strands of DNA after they've been unwound from those histone proteins, right? It's then going to add the complementary nucleotides to a new molecule of mRNA, right? And then each gene ends with a three-letter codon that signals to stop, to end. We're done, right? You've reached the finish line. Then that is going to cause everything to detach here. right? So we've got our pre-mRNA, right? We've already said we got to remove all that that intron data, right? Spliceosomes. That's the most ridiculous name I've ever heard, but it is what it says on the tin, right? It's an enzyme that cuts and splices the mRNA to remove the intron data, right? So again, it's meaningful, even if it sounds a little silly. And of course, we have self-check mechanisms, right? Our DNA and RNA production mechanisms inside the cell have a lot of proofreading and error checking mechanisms involved so that we transport them accurately. So taking a look here, I'm going to zoom in. Right. So we have a DNA, a promoter region, right? This is some of the intron stuff in the DNA, right? We need it there so that this enzyme knows where to attach and where to start working. And so that is going to bind to the promoter region, open up another replication bubble right here, and initiate mRNA synthesis. Now, unlike DNA replication, mRNA synthesis is only using one of the strands of DNA, right? We call it the template strand, right? We're only making one copy, so we only need to read one strand. And because it's a lot more efficient, right, we are going to read the DNA on the strand that follows the same direction that we're unzipping it, okay? So we don't have any of those Okazaki fragments or anything like that. Now that just continues, right? That's elongation. It's not really all that complicated, right? It's like a zipper moving down your coat, right? It opens it up on one side and closes it back on the back end, keeping everything nice and tidy. And then finally, once we reach that stop signal, right, then we detach the mRNA transcript is released from the RNA polymerase. And this enzyme may go on and it may make thousands of copies of that particular piece of DNA and transcribe it into mRNA depending on the needs of the cell and what's going on. If we zoom in even further, right, the template strand is where we are attaching those new nucleotides, right, to make our mRNA. Now, again, because they are complementary to the template strand, they are going to match the code exactly of the other strand, right? Because the other strand of DNA is also complementary to the template strand, right? So if mRNA used the same four bases as DNA did, right, and we didn't replace thymine with uracil, the code of the mRNA and the code of the coding strand would be identical, right? But because mRNA does use uracil instead of thymine, it's identical except that where we see thymine in the coding strand of DNA, we will see uracil in the mRNA. I feel that goes by a little quickly sometimes, right? So the template strand is what we're reading, and that's what we're matching up to, right? Plugging in complementary nucleotides. The mRNA is going to be the same code, the same sequence of bases as the coding strand of DNA, right? The part that we're not really reading. It's that leftover part, except for we're switching out the thymines with uracil. Okay. I need to unzoom before it'll let me advance. So this is the same stuff it's the same, literally the same picture, just in pieces. So we'll go ahead and move on from there. How am I doing on time? About out of time. Okay, so that's the big that's the big idea for transcription. Translation is its whole thing. I'm out of time. Watch the video, right? Watch the video. Um, knowing the what is involved with the different subunits of the ribosome, where the ribosomal RNA is involved, the different binding sites in the ribosome that allows the mRNA to be kind of moved through the factory and plug in the amino acids, right? How the transfer RNA is involved. That's all very important. You need to know all of that. Okay. Send me your questions. Send me your questions tomorrow after you go through that. Cool. I'll meet y'all over in lab in about 7, 8 minutes. I got through more of that than I thought I would, if I'm being honest.